So welcome you back, my dear students. Hope you are doing well and are very safe at home at this pandemic situation. So now in this video lecture tutorial, we will talk about preparation of competent cells for transformation. So already we have discussed about transformation. Transformation means the insertion of short piece of DNA into bacterial cell is called transformation. And competent cell means whether the cell are able to take up the foreign DNA material or not, how much the cells are competent, that you have to decide, means you have to prepare. Okay, now the cells are competent, we can insert foreign DNA material. So that's why competent cell is very important for transformation. So introduction of the DNA into host cell, there are mainly two key problems. Must be able to physically cross the cell membrane, and once inside the new host cell, our DNA must be able to replicate. So this is the two thing you need to keep in your mind. Must be able to physically cross the cell membrane means, so competent cells should be selected based on these two things, like the cell membrane should be very thin so that the DNA material can be easily inserted, and that DNA must be able to replicate inside the cell. That environment should have these two pointers, very important. So now transformation is already discussed about, is the genetic alteration of a cell resulting from the direct uptake of and incorporation of exogenous genetic material, means the foreign DNA material from its surrounding cell, the cell membranes. Okay, so for transformation to take place, the recipient bacteria must be in a state of competence. Here is the thing, competence means the bacteria should be competent, should be able to take up the DNA material. Okay, so which might occur in nature as a time limited response to environmental conditions such as starvation, cell density, and these other things. So now, what are the actually competent cells? Bacterial cells that are able to take up DNA from the environment are called competent cells. So they are competent. So means, suppose you are competent to take foreign material in your body. So likewise, the bacteria are also competent then only when you can able to insert the foreign DNA material inside the cell. In the laboratory, bacterial cells can be made competent and DNA subsequently introduced by a procedure. So in the laboratory conditions, lab condition for our experimental needs, actually, we can develop the bacterial cells into a competent cell. Okay, so there is a protocol. Now we will discuss the protocol. Methods of transformation is basically the cells are naturally competent use is bacteria Bacillus subtilis, and there are several methods of transferring the DNA material into bacterial cells. That is the calcium treatment of the cell, transformation of the protoplast, and electroporation cells of the or electroporation of the protoplast. That is nothing but using the high voltage of shock. Okay, so this is the type. So very popular and common type is the calcium treatment of the cell. It's very easy and very high frequency level is very high. The competency level is very high in case of calcium treatment of the cell. And there are another procedure, micro injectile bombardment injection of the cell. Is the, you can remember the gene transfer method. This is the gene transformation, micro injection through the gene gun. You can directly insert into the nucleus the genes. So there are some limitation, advantage, and disadvantages. But in case of calcium chloride treatment of the cell, competent cell is very popular. And the heats of transformation is also very important. Heats of transformation is effective. Heats of transformation used in calcium rich environment provided by calcium chloride to counteract the electrostatic repulsion between the plasmid DNA and bacterial cellular membrane. Okay, a sudden increase of temperature creates pores in the plasma membrane. So plasma membranes in the cell membrane of the bacteria and allow to plasmid DNA to enter into the bacterial cell. So calcium chloride and heat, they generate this kind of environment, electrostatic repulsion between the plasmic DNA and bacterial cellular DNA, which makes a pore in the plasma membrane and can insert the plasmid DNA into the bacterial cell. So now we are discussing about the transformation using calcium chloride. So what are the things material required? The buffers and solution is calcium chloride, 1 molar, standard transformation buffer, TFB is the magnesium chloride or calcium chloride solution, and cold ice. And what are the media is required? Media, why media is required? Because you need to culture the transformant, means the transformed bacterial cell, and LS media or LB media or SOB media. Okay, so these are the, or SOC media or LB. LB is the very popular media, Luria Bartoni. Okay, so LB media here it is not mentioned, but LB is the very popular media. You can prepare for your computational experiment. And nucleic acid or oligonucleotides, plasmid DNA, centrifuge and rotor. These are the materials you need. Okay, Sorval GSA rotor or equivalent is the specific. And equipments, what are the things you need? Polypropylene tube, 50 ml, chilled in ice. Okay, and water bath preset at 42 degrees centigrade, because in the procedure you need heat shock at 42 degrees centigrade. So now we are coming to the procedure. So procedure already I have given in the notes, so just I am highlighting here the points. Incubation of the E. coli culture into the LB medium. See, here is the LB medium, and incubate in 37 degrees centigrade for 1 day with vigorous shaking on at 80 RPM. Okay, then alicot was taken into a 50 ml LB media and pre-warm broth to, so incubation at 37 degrees centigrade, shaking on 80 rpm. Monitor the growth regularly. OD check the OD, and it should be in between 0.35 to zero point 0.4. Okay, so that means is the good growth. When suitable growth has been reached, chill the culture on ice. Then the transfer culture and an autoclaved centrifuge tube and collect the cell pellets by centrifugation 6000 rpm for 5 minutes at 4 degrees. So at the chill centrifugation for 5 minutes, this is the rpm. You just centrifuge the cell suspension and it will make a pellet, the cell, and resuspend the pellet means at the bottom of the centrifuge tube with resuspend this pellet 20 ml of ice cold 50 millimolar calcium chloride solution. So incubate the resuspended cell and ice for 20 minutes. So collect the cell pellets, centrifuge again. The cell pellets will be there, and with the cell pellets, you need to 2.5 ml of ice cool again calcium chloride suspension. You just dissolve it, and finally the 10% glycerol you have to add to make all the cells are viable. Then use 100 microliter of the prepared competent cells for transformation. So this is the very important and crucial point. Dispense the competent cells into alicot of 100 microliter and store them at minus 80 degree for further use. For future experiment, you can keep the competent cell, making the alicot in several tubes of 100 microliter and keep it at minus 80 degrees centigrade for further use. And initially, you may take 100 microliter for transformation. So here in the conclusion part, we can tell you non-commercial preparation should normally give 10 to the power 6 to 10 to the 7 transformation per organism of plasma. And this is the normal range. A poor preparation will be about 10 to the power 4 per microgram or less. But a good preparation competence can give up to approximately 10 to the power 8 colonies per microgram of per plasmid. So that means this microgram is nothing but the concentration and volume, you can say, of plasmid DNA, the foreign DNA you want to insert. So protocols, however, exist the making super competent cells that may yield transformation efficiency over 10 to the power 8. So actually, if you get up to 10 to the power 8 colonies per microgram of plasmid insertion, because once the competent cell is prepared, you can insert the competent cell with the plasmid DNA, add the plasmid DNA into the competent cell and heats for 90 minutes, 90 seconds at 42 degrees centigrade temperature. Okay. So the chemical method, however, usually does not work well for the DNA. So this is not so important. So here is the important thing: precautions. What are the precautions? Means you have to take care of because calcium chloride is a hazardous material for skin, okay, eyes, and the respiratory system. So it may cause burns. Hence, use gloves while using the same. Okay. So gloves is very important. Avoid thawing the cells before use. So this is when you are bringing it for minus 80 degrees centigrade. So thaw the cells before use. Keep cells on ice not longer than 3 hours. Do not use cell again that have been on ice. Okay. So you may stock freeze the competent cell in liquid nitrogen or stock freezing might keep cells viable for a longer period but decrease the transformation efficiency. But so I hope you can understand what is the actually thing. So this is the overall procedure. Just in the slides you have seen only the theoretical part. Now in the picture you can clear the whole thing. This is your E. coli culture. This is the competent cell. You are culturing on LB broth medium. Okay. Then centrifuge. Centrifuge means 6,000 RPM for 5 minutes at 4 degrees centigrade temperature on a 50 ml culture tube or centrifuge tube. Then the pellet will form. The pellet will be suspended, resuspended with calcium chloride solution. Then chill on ice. Okay, so repeat this experiment, this step 2 times, because once only calcium chloride, then only calcium chloride plus 10% glycerol, because you are making stock for your further experiment. So this is the alicot. You just you have a lot of volume. You just taken in several Lappendorf tube and insert. This is your experimental sample, and these things you can keep it for further use and keep it at minus 80 degree. And this is your competent cell. You are inserting and you are inserting ampicillin resistant plasmid here. So you have added chill on ice, then heat soak. Here, see, 40 degree centigrade for 90 minutes. After heat soak, plate on LB plus ampicillin resistant, because this here is the AMP ampicillin R means here is the R, ampicillin R means ampicillin resistant gene. So these colonies, if you add the LB medium with ampicillin, because the plasmid is there already, they can resist the ampicillin, so that they can grow, they should grow actually. So once the colonies are growing on the LB plate, so you can claim these cells are competent, because already you have inserted ampicillin resistant gene, and in the media ampicillin is there, but the cells are growing. That means the cells are resist to ampicillin. So your transformation is successful. Hope you can understand in this video, and let us know if you have any doubt. We will take care of this. Thank you.